So after studying this module, you shall be able to learn the concept of motivation and learn about the hierarchy of motives, understand the role of homeostasis as the basis of physiological motives, and learn about the hormonal and neural basis of some of the important motives such as hunger, thirst, sexual motivation, aggression, and psychosocial motives in the light of reinforcement processes. This module introduces you to the various types of motives and the role of brain structures in different motives. According to Gehrig and Zimbardo in 2009, motivation is a general term for all the processes involved in the starting, directing, and maintaining physical and psychological activities. The word motivation comes from the Latin word movere, which means to move. All organisms move toward certain stimuli and activities and away from others as dictated by their appetites and aversions. Motivation is not a concrete thing. It is an abstract term which can be inferred from the behavior of organisms. If a person is angry, we will not get to know this until he shows it in some way. If our inference about the motive is correct, we can also explain and predict the behavior of organisms. This gives us an idea about the whole range of things the person will do. Motivational states are geared towards the survival of organisms. Let us now understand what is homeostasis and motivational states. The basic requirement for survival is the state of homeostasis. Several mechanisms involuntarily work to keep various chemical and other factors of the body in a fixed range. Going significantly away from this range means disease and death. The basic conditions of hunger, thirst, temperature regulation, and sexual motivation, etc. need to be considered in this context. Physiological motives arise due to imbalances in the physiology of the body systems. The body maintains a balanced state called homeostasis. When the organisms depart from this homeostatic state, there arises a need to restore this balance, and thereby the physiological motives are triggered. For example, when a person is feeling hot, the body is moving away from its normal temperature of 98.4 degree Fahrenheit, and therefore the physiological motive or need to balance this temperature arises. In response, the body first shivers to cool itself, and later, if homeostasis is not achieved, the person reaches out to cooler or air conditioners. Here, the shivering of the body is automatic physiological mechanism aroused to achieve homeostasis, and reaching out to coolers and air conditioners are behaviors that the person voluntarily indulges in to restore the bodily balance. Not only the deviation from homeostasis, other triggers are the arousal of physiological motives or states. In certain species of animals, hormonal release triggers the physiological need for sexual intercourse. Pain stimulus, when sensed by humans, triggers the physiological need to avoid its source. Many theorists have tried to explain the concept of motivation. Maslow in 1970 gave the hierarchy of motivational needs. The basic concept is that a motive higher on hierarchy can be achieved only after the lower ones have been achieved. The Maslow's hierarchy of need is depicted in the diagram, starting from biological need, then to safety need, then to attachment need, then to self-esteem need, and finally the self-actualization need. Maslow concluded that the fulfillment of need takes place in a hierarchy. The needs higher on the ladder takes their precedence only after those lower on it are satisfied. A hungry individual cannot be driven to self-actualization. He must satisfy his hunger first. All physiological needs are the first priority, since they are most linked with the survival of the organism. Let us now study the first motive, which is hunger. Motivation of hunger is necessary for survival. Food provides energy to live and for the proper functioning of the body. Several questions need to be answered by a physiologist, such as why do we feel hungry? What brain areas are involved in the process? And when and why do we stop eating? Early researchers have answered these questions by performing experiments. Hunger has been described as a chemically controlled process and or controlled by the contractions of the stomach. Later researchers suggested the role of hypothalamus in regulating the stomach contractions as well as the chemical processes to explain the hunger motivation. In an early attempt to find the causes of hunger motivation, Cannon conducted an experiment and showed that the gastric activity in an empty stomach was the sole basis of hunger. In his experiment, the subject swallowed an uninflated balloon attached to a rubber tube. The other side was attached to a device which measured changes in the air pressure. Then the balloon was inflated inside the stomach. As the subject's stomach contracted, air was expelled from the balloon and activated the recording device. These reports were correlated with the periods when the subject was hungry, and the record showed that his stomach was severely contracted. This experiment led Cannon to prove that the stomach cramps were responsible for hunger. Later researchers confirmed that stomach contractions were not even a necessary condition for hunger. According to them, level of use of dissolved blood nutrients is responsible for hunger motivation. They focused their attention on glucose and fatty acids. They correlated low rate of glucose with the reports of hunger and eating behavior and high utilization rate with the reports of satiety or absence of hunger motivation. The role of fatty acid is just being investigated. The primary focus has been on the brain areas involved in the whole process. The stomach conveys satiety messages to the brain via the vagus nerve and the splanchnic nerves. The vagus nerve conveys information about stretching of the stomach walls. Animals with a damaged vagus nerve eat until they overfill their stomach. The splanchnic nerves carry impulses from thoracic and lumbar parts of the spinal cord to the digestive organs and carry information about the nutrient contents of the stomach. The role of hypothalamus has been found to be the most significant in this context. The classic work of the 1940s and 50s emphasized the role of hypothalamus by either stimulating it or removing it. They found the role of lateral hypothalamus or LH in exciting the region of hunger motivation and the role of ventromedial hypothalamus VMH in satiety. Electrical stimulation of LH was found to elicit eating. VMH stimulation was found to stop ongoing eating behavior. The lesions made in the two areas were found to have effects opposite to those of stimulation. These studies used animals to study the effect of lesion and the stimulation of LH and VMH. Animals with damage to the LH would not eat or drink and eventually died of starvation unless given special care. It was found that rats with ventromedial lesions developed voracious appetites, consumed great quantity of food and gained weight rapidly. Besides VMN, the paraventricle nuclei of hypothalamus also plays an important role in hunger. It may contribute to diet selection as well as to ending of the meal. An injection of norepinephrine on PVN increases carbohydrate intake, whereas one of aldosterone or the neurotransmitter galanin will increase the fat intake. One complication in the interpretation of lateral hypothalamus as area controlling hunger is that in lesion studies, some dopamine controlling neurons also get damaged, which pass through the lateral hypothalamus. Such damage leads to the lack of arousal and active search for food, even though if given food, such animals may eat normally. The role of lateral hypothalamus in feeding behavior is multifaceted. First, axons from here extend to the nucleus of the tractus solitarius in the pons, which increases salivation and taste sensation. Second, it stimulates the forebrain structures, facilitating ingestion and swallowing. Thirdly, it stimulates the pancreas to release more of the insulin and digestive juices to facilitate the process of metabolism. According to Morgan and King in 1993, the results of these studies indicate that the LH is a feeding center, while the VMH is a satiety center. Recent studies have confirmed these findings, but have raised questions about the interpretation of the LH and the VMH as the only centers for the control of hunger derive. First, other brain areas such as amygdala play an important role in hunger and eating. Second, the LH and the VMH effects on eating are due wholly or in part to what happens in the nerve fiber pathways which pass through the lateral hypothalamic region or the ventromedial region. Third, at least some of the effects of the lateral hypothalamic lesions seem to be due not so much to a decrease in hunger motivation, to lower levels of arousal and general neglect of sensory inputs, but to the stimulation of other neural centers as well. The current view is that the classic hypothalamic centers may be more accurately considered to be parts of the brain systems involved in monitoring the body's fuel supplies, controlling metabolisms and perceiving food related stimuli. The role of the hormone CCK or cholecystokinin, which the duodenum releases and the brain uses as a neurotransmitter, has also been highlighted to end the meal taking behavior or to indicate satiety. Introducing food into the duodenum releases CCK. When CCK is injected into the duodenum, the eating behavior of the animal is drastically reduced. Let us now come to the next motivation, that is thirst. Another basic motivation is thirst. Water is very essential for living. Water level as well as the chemicals present in water are necessary for homeostasis. The body has set up a very complicated mechanism to regulate its fluid level and drinking behavior. There are some hormones which help in regulating the body fluid level, like the antidiuretic hormone or ADH, but it cannot directly regulate thirst without the control mechanisms in the brain. Drinking occurs for many reasons. Most drinking occurs around meal time when stomach is stimulated by food. An increase in the osmotic pressure of the blood, which draws water out of the cells, causes osmotic thirst. Neurons in the organum vasculosum laminae terminals or OVLT, an area adjoining the third ventricle, detects changes in osmotic pressure and sends information to the hypothalamic areas responsible for vasopressin or an antidiuretic hormone and initiate drinking. There is another convincing cause of thirst motivation caused double depletion hypothesis. According to this hypothesis, thirst motivation and drinking are mainly triggered by two conditions of the body, that is, the loss of water from cells and reduction of blood volume. When water is lost with body fluids, it leaves the interior of the cells, thus dehydrating them. In the anterior hypothalamus, there are nerve cells called osmoreceptors which generate nerve impulses when they are dehydrated. These nerve impulses then act as a signal for thirst and drinking. The thirst triggered by the loss of water from the osmoreceptors is called cellular dehydration thirst. Loss of water from the blood also results in hypovolemia, that is, decrease in the volume of the blood. This results in the drop of blood pressure, which stimulates the kidneys to release an enzyme called renin. The renin, through a several step process, is involved in the formation of a substance known as angiotensin II that circulates in the blood and may trigger drinking. However, there are questions raised about the role of this hormone in drinking, as other mechanisms are also involved. The loss of blood volume causes hypovolemic thirst. Animals with such thirst drink more water containing solutes than pure water. The subfornical organ is especially important for detecting changes in blood volume and sending information to trigger hypovolemic thirst. The two stimuli for this thirst are baroreceptors and the hormone angiotensin II, which increase when blood pressure falls. They act synergistically. Loss of sodium salts from the body also triggers thirst because with salt one tends to lose water also. The hormones aldosterone and angiotensin too stimulate such cravings. Let us now understand what is sexual motivation. Like other motivation, sex is a physiological need and it strongly drives behavior to satisfy it when the animal is sexually stimulated. There are other aspects attached to sexual motivation other than physiology. Also, there are characteristics that separate sexual motivation from other types of motivations. First, sex is not necessary to maintain life of an individual, although it is necessary for the survival of species. Second, sexual behavior is not aroused by a lack of substances in the body. Third, in higher animals, sexual motivation is mainly due to sensory information from the environment, including incentives, than are only physiological drives. Sex hormones in the body are the major physiological source of sexual arousal or motivation. The sex hormones are released by sex organs and adrenal gland. Hypothalamus, particularly the preoptic area, plays a major role in regulating the release of such hormones. The organization of hypothalamic structures and their role in the release of sex hormones is very complex. Female sex hormones are estrogens and male sex hormones are androgens. However, both these hormones are present both in the females and males. It is determined by the quantity of these hormones present in the body whether the person is male or female. Depending on the genes of the fetus, female sex organs, ovary or male sex organ, testis, develop in the fetus. Later, at the time of puberty, sex hormones are released greatly from the sex organs, which results in the development of secondary sexual characteristics. These secondary sexual characters differentiate between female and male, and also they act as external stimuli to trigger the sexual motivation in both. So this is clear that hypothalamus plays an important role in the regulation of sex hormones in both females and males. The hypothalamus controls the activity of the pituitary gland by nerve impulses and by releasing hormones. The pituitary in return secretes hormones that alter the activity of the sexual hormones. Sexual hormones also exert organizing influence on the other aspects of behavior. In adulthood, sex hormones can trigger sex behaviors, aggressive behaviors or general activity levels. Such behaviors are affected by the factors of biological and physiological origin. The position of the hypothalamus, adrenal glands and sex organs in female and male body are depicted in the picture. Now, question arises is how actually organisms get motivated for sexual behavior, or does the increase in the hormonal level trigger sexual behavior? The answer is yes, for the organisms of lower and some of the higher species. In females of lower species, higher levels of estrogen level in the blood activates the sexual behavior, which is called estrus or heat. In human females, there is a contradictory evidence about the activation of sexual behavior from the increased level of estrogen. Some studies have shown that the sexual behavior increases at the midpoint of the menstrual cycle, when the level of the estrogen is high. Other studies have shown that sexual behavior increases at the time just after the menstruation. However, some other studies have found that the sexual behavior decreases after the menopause when the level of estrogen decreases. These studies show that the level of estrogen and sexual behavior are related. In males of lower level species and humans, the activation process of sexual behavior is alike. There is clear evidence that the increased level of androgen and especially testosterone triggers the sexual behavior in males. The threshold level of testosterone is enough to activate sexual behavior in males. The level of estrogen above the threshold level does not activate the sexual behavior more and more. It is just the threshold level which is enough to activate sexual behavior. The increase in the amount of sex hormones just activates the urge of sexual behavior, but it is not a compulsory condition to indulge in sexual behavior. The actual sexual behavior is triggered by the presence of external stimuli. for example, the presence of sexual partner. There is still a lot of variation in the activation of the sexual behavior when the hormonal levels are appropriate. Let us now come to the next motivation, that is aggression. Aggression is the harm done consciously to another person or to own self. Like sexual motivation, aggression is neither a necessary condition for life nor it is due to lack of some substance in the body. However, it has a great adaptive and survival value when the need arises. It has been found that the brain is greatly involved in the causation of aggression. Hypothalamus, in association with the other brain parts, is responsible for the aggression motivation. Studies show that the hypothalamus and the periaqueductal grey matter of the midbrain play a critical role in aggression. Electrical stimulation of the hypothalamus lead to aggressive behavior. There are receptors in the hypothalamus which determine the level of aggression based on their interaction with the level of serotonin and other neurotransmitters in the brain. Amygdala is also associated with the regulation of aggressive behavior. Many studies with lesion of amygdala show that the organism's behavior to compete and to dominate decreases. The role of amygdala in primates is not very clear. It has been observed that the lesion of amygdala either results in increased social affiliation or aggressive response. The picture highlights the position of prefrontal cortex and amygdala in the human brain. The role of prefrontal cortex is seen in regulating aggression. Reduced activity of the prefrontal cortex results in the violent and antisocial activity. The role of chemicals in the brain, particularly neurotransmitters, have been studied. A deficit in serotonin has been found to have primary role in causing impulsivity and aggression. Nevertheless, low levels of serotonin transmission may explain a vulnerability to impulsiveness, potential aggression, and may have an effect through interactions with other neurochemical systems. These include dopamine systems, which are generally associated with the attention and motivation towards rewards and operate at various levels. Norepinephrine may influence aggression responses both directly and indirectly through the hormonal system, the sympathetic nervous system or the central nervous system. Similarly, GABA, although associated with inhibitory functions at many central nervous system synapses, sometimes show a positive correlation with aggression, particularly when potentiated by alcohol. Hormones like testosterone have been linked on average to more physical reaction in many species. The increase in the testosterone levels lead to increase in aggressive behavior in humans also. These findings indicate the sex difference in the sexual behavior. The level of testosterone is less in females as compared to males, resulting in less physical aggressive behavior in females as compared to males. In lower animal species, it has been seen that the testosterone levels increases in males when competing for females for mating. At this point, they need to be aggressive to compete with other males. But this kind of behavior is not observed in human males. Let us now understand what are psychosocial motives. Some psychosocial motives are governed by hormones. The motive of nurturance and maternal behavior are examples of that. They are governed by the hormone oxytocin. Other such motives have evolved since they have evolutionary and survival advantage. Protection provided by living in a group has given rise to the motive of gregariousness. They are thus based on the principles of reward, punishment, and the reinforcement process. Several brain areas contribute to that. The behavior found useful and providing positive reinforcements are retained. The signal in the brain usually travels from the cerebral cortex that signals that a stimulus in the external environment is beneficial and thus the behavior of the self is reward worthy. An area called the ventral tegmental area or VTA receives signals from the cerebral cortex and releases dopamine into the nucleus accumbens, amygdala and the prefrontal cortex. Stimulation of the nucleus accumbens in humans elicits smiling, laughter pleasurable feelings, happiness, even euphoria, and is thus a positively reinforcing stimulus. This area of the brain, called ventral tegmental area, amongst the most primitive parts of the brain, synthesizes the main neurotransmitter substance that is implicated in the reward circuitry, pleasure centers of the brain. It is the neurotransmitter called dopamine. The brain structure is located at the top of the brain stem in the midbrain. Amygdala and its connections to the prefrontal cortex and basal ganglia are likely to influence the selection and initiation of behaviors aimed at obtaining rewards and avoiding punishments. Well students, now let us summarize what all we have learned. First of all, that the basis of motivation is to maintain homeostasis of the body. Motives exist in a hierarchy from the simple physiological to the most complex self-actualization. Next, lateral hypothalamus and ventromedial and paraventricle nuclei of the hypothalamus are involved in the motivation of hunger and thirst. Neurons in the organum vasculosum laminae terminals or OVLT, an area adjoining the third ventricle, are involved with drinking. Sexual motivation is controlled by the hormonal secretions and the hypothalamic areas. The role of amygdala is important in the context of aggression, and the physiological motives are controlled by hormones like oxytocin, several neurotransmitters, and brain areas like limbic system and prefrontal cortex, among others.